So instead of me telling you, you tell me what's on your third test.
Cranial nerves.
Cranial nerves, just that little bit, okay? ANS and the rest of the PNS, right? Spinal cord, spinal nerves. Okay, that's one set of notes. Heart, blood vessels, yeah? That blood is easy. It's not really, what? Heart is easy, I think. I think vessels is easy, honestly. I don't think any of that stuff is complicated. Are you guys doing okay on vessels and lab? Yeah, after you like know where to expect it, you're like, yeah, of course that's called the brachial artery. What the hell else would you call it, right? So luckily that happens, and you've got a lot of confidence by this time with the cadavers, so that you can kind of figure out things in context. So that shouldn't be bad. FYI, on this test, the thing that people generally have the most trouble with is the nervous system stuff. They can't remember nervous system terminology, so I'm telling you this so that you won't be one of those people. They don't remember somatic motor, autonomic motor, visceral sensory, all of that kind of stuff. And remember that even though we're not testing from that terminology, you have to use it again, right? In this stuff, you have to talk about it again when you're talking about how the spinal cord works, motor versus sensory, and all of that kind of stuff. So make sure that you brush that off. The terminology associated with the autonomic nervous system, everybody flippin' hates it. It is sympathetic, parasympathetic, preganglionic, postganglionic, all of that business, inhibit, excite, you know, whatever it's doing to everything. So you still got some time. You got until Tuesday. That is the stuff you are probably going to need to be most critical of yourself about when you're studying, right? It just doesn't stick very well. I don't know why. It didn't stick very well for me either. So as far as lab goes, you are in the home stretch, right? So you are starting this Friday, respiratory and digestive, and then you've got one more lab after that, which is urinary and reproductive, and then your lab practical. So let's talk about that. Your lab practical is the same week as your last lecture test, okay? There's a lot of overlap in material, which is good. Your last lecture test is urinary, reproductive, and lymphatic. So you don't have lymphatic in lab, but urinary and reproductive. You don't have an extra week to study like you did before the midterm. And then the other thing that happens is that your lab practical, your lecture test are in 1 week, and then your lecture final is the next Thursday. So I'm telling you guys this because, you know, planning out your time over these last few weeks gets problematic if you let it pile up on you, and I would prefer that you guys do well. Go ahead.
So if we have an A in the class, by that point, do we email you? Or do you, so like, we just don't have to show up to the final?
No. Well, let's talk about that final and A thing, okay? So here is a bit of information that can help reduce stress. First, remember that if you are making a 92.5 or above, I will round up to a 93, and that's an A. And philosophically, you can't get A'er, so I don't know why you have to take the final. So you can stay home and feel good about yourself. But that depends on your lab instructors getting their grades to me. So you are going to have to be kind, persistent, bring them treats, include lots of emails that say the word please and have lots of appreciative things happening. Otherwise, they won't get the grades to me, and so you won't know. But worst case scenario, you don't know. You come take the final. But here's the deal about the final. I've arranged it so that it encourages you to continue trying. But the final literally cannot hurt you if you have taken all 4 of your lecture tests. Because if you take all 4 of your lecture tests and your final ends up lower, I will drop it. Okay? So you will get a minimum of whatever grade you got going into the final, and it can go higher if you have taken all of your lecture tests. So I just want to keep you trying until the end. If you haven't taken all your lecture tests, your final can hurt you, right? You can actually drop your grade. But other than that, so if you get into a situation in which you don't know if you're making an A, you should go take the final. There's nothing that I can do to get your lab instructors to get their grades to me faster. The reason that this happened, that it's kind of squished at the last end, at the end here, is because we gave you that extra week for studying before your midterm. That's why lab goes longer. So it was, had to balance it out, and the time has to come from somewhere. They do try, though. Okay, so everybody pretty clear about everything that's going on. In addition, consider, for those of you who like anatomy, for those of you who are really going to be freaking glad when it's over, don't listen to this, but for those of you who like anatomy, consider for fall doing the lab aid class, which is Biology 2327. It's a lot of fun. There are no tests. There are no quizzes. There's a lot of conversation about the best ways to learn and the best ways to teach, and you get to do a cool project. Some of the projects have ended up being presented at scientific meetings all over the country. We've got one that's probably in for publication, and they'll get an undergraduate publication before they graduate. In addition, it's a lot of fun. And if you teach something 20 different times over a week, you are literally going to know it way deeper than you ever could have by studying it out of a book. So if you are the kind of person that thinks that would benefit. In addition, most of my lab aids, it looks really, really good on applications for things. I've got 4 lab aids who are done with nursing school. I've got 2 who are in their last semester of their DPT programs. They're about to finish physical therapy. I've got 2 that are in med school. These guys generally do rather well. They also, it builds a lot of confidence by being able to talk about that stuff with multiple people, and it's kind of fun. So if you're curious about it, you can send me an email and I'll tell you about the class, or just ask one of the lab aides. They'll tell you. They'll tell you whether they like it or some of them just continue to come back to do it multiple years. I've had somebody who's been volunteering to do it for 4 years after she took the class. So, all right, so let's finish blood. And then we will briefly start on lymphatic system, and then I'll let you guys go. Okay, so only thing we had to do here was platelets. Okay, so let's talk about platelets and where they fit into the story. First, remember that when you were looking at a hematocrit, 44-ish% red blood cells, 55-ish% fluid, plasma, and then 1% collectively is the leukocytes plus the platelets. Platelets are far more abundant than the leukocytes. In a cubic millimeter, you had 4 to 6 million red blood cells, 4 to 11,000 white blood cells total, all 5 times. And then this one, it says 150 to 400, but it's usually listed as like 250 to 400 or 450. So it's a little bit variable. So when you look at a blood smear, by the way, you are generally going to see platelets. They're way smaller. You're always going to see red blood cells, of course, because they're so damn many. And sometimes you'll see white blood cells, but you will always see platelets. Almost always. So you can usually see at least one. They're smaller than red blood cells. They don't look like much because they're actually not whole cells. So let's look at how they are actually formed from this figure that we looked at last time. Again, you're not responsible for the details, but they come from myeloid stem cells, and what happens is you develop this enormous cell called a megakaryocyte, and then the megakaryocyte busts into little pieces, and what platelets are little membrane-filled bags of clotting proteins. They're not whole cells. They don't have a nucleus. They can't go through cell division. So that's why collectively you call all of those things formed elements, although before they knew they weren't whole cells, they named them thrombocytes. But they're not whole cells. It's just sort of an anomaly before they actually knew. Okay, so these are, they have no nucleus. They only live about a week in circulation. Their primary function is to initiate a chain of reactions that results in blood clotting. They are not responsible for blood clotting alone. When you get into physiology, you will see that blood clotting is a pretty damn complicated proposition, but platelets are always involved in it. Go ahead.
Do the megakaryocytes break themselves up, or is it mediated by, I guess, the
actually don't know the answer to that question. No, it's a good question. It just never occurred to me to ask before. If you think about it, you can email me, and then we'll both know, and I can teach you guys. But you know I won't remember unless you email me. We know this about me, right? I'm a very curious person with like the attention span of a gnat or a puppy. Like, oh, shiny object, what's happening? But I love doing research too. I love it. Like, I love reading all the original literature. And I was so incredibly happy when I figured out that my 10-year-old, he did his first research project, and he was like, oh my God, Mom, I love doing research. It's like the most fun thing. I'm like, oh my God, it's the best. He's like, where can I find more information? I'm like, I know, this is my life. Okay, so let me show you what a clot looks like. This is a type of microscopy showing you what a clot looks like, which is kind of cool. This is scanning electron microscopy. So the deal with clots is that it's initiated by platelets, but the actual clot is not made of platelets. The actual clot is made of this gooey, long insoluble proteins called fibrin. And fibrin, think of it like spiderweb fibers, because it's really strong collectively, but it's also kind of sticky. So what happens is it catches everything that's going by in the fibrin. And the objective, of course, is to plug a hole. So you poke a hole in a blood vessel, and what the clot is supposed to do is to stop you losing fluid through the hole, right? And so that's what fibrin does. The erythrocytes are really not part of it either. They just got caught, like, like flies in the spider web. But the other thing is that in a healthy person, when things are going well, after the fibrin has plugged the hole for long enough for the hole to be repaired, there are mechanisms to dissolve the fibrin so that you regain blood flow. Does that make sense? So what happens if you can't clot normally? Well, if you can't clot normally, then of course you're going to run the risk of bleeding out if you ever get injured. There are a bunch of different reasons for that. One of them is hemophilia, right? You guys have heard of hemophilia. Hemophilia is generally genetic. It means that there's a whole bunch of factors involved in clotting. If you lose one of them in a crucial point in the pathway, and that's what generally hemophilia is about, then you can't clot. It's actually not a platelet problem. It's one of the other factors. And you probably heard of hemophilia with like the royal family. It's factor 8 hemophilia. There was just like one little genetic factor. And it wouldn't have been much of a problem, but they kept like marrying their first cousins and all of that kind of stuff. So that became problematic. And you had a whole bunch of like royalty die because of that. And it was really just because that they thought that that the royal blood was special blood. And so they had to marry.
Was it on the X?
The type of hemophilia in the royal family was carried on the X chromosome, so far more males were affected. Not all forms of hemophilia are like that, but that form of hemophilia was like that. So inbreeding, not so cool. Even when you're super rich.
Cool, but
It seems cool, it actually doesn't. There are all kinds of evolutionary protections against inbreeding. It's really funny. This is just an aside, but hey, we're like 25 minutes ahead. So my background is in evolutionary biology before I went into molecular biology, and one of them is that mammals specifically, other organisms too, but mammals specifically have all kinds of protections against inbreeding. And one of them is simply that testosterone, when you start getting high levels of testosterone in males, it tends to make you roam, right? And when you roam, you're far more likely to run into someone who's not your sister, right? It increases risk-taking behaviors and all those kinds of things. And the second thing is that you tend to be repulsed sexually by scents that are too much like yours, right? Too much genetic homology. Like, I mean, it's super gross to think of even now. Like, when people tell you that your brother is incredibly sexy, you're like, that is the most repulsive thing I've ever heard of. Because you're like, oh my God, what the hell? Even though he looks like mostly like you and you think you're pretty good looking, you're like, get anywhere close to him and like everything about you just dries up and you just like want to punch him. That's an evolutionary advantage. It's a protection against inbreeding. And it's apparently because of like the scents that you release, and the scents that you release are too much like you is aversion. What were you going to say, Charlie?
So there's a mental disorder or something like that
back together if they have like, a romantic relationship.
Oh,
Yeah, something like that. And so
I don't know what the hell's going on there. Well, I mean, nothing is 100%, right?
Yeah.
Yeah? Yeah, I don't know. I know that it's pretty damn effective because, of course, we have far less inbreeding than you would imagine, especially since we tend to stay in groups, right? We tend to stay in groups, but there's not that much inbreeding. In addition, of course, after you got societies involved, then there's like all kinds of social prohibitions against inbreeding, unless you're royalty.
Or the Ozarks.
the royalty can
In the
afford
Ozarks.
expensive
What? Yeah, expensive perfumes. Yeah. Okay, I don't know why I started talking about that. Just because it did. Okay, so let's talk about when you clot inappropriately and why you clot inappropriately. Inappropriate clotting in an unbroken blood vessel does occur, and that is called thrombosis. So the things that increase your likelihood of thrombosis are whenever platelets congregate, they like to start the clotting process. So I always say that platelets should not congregate unsupervised like teenagers, right? They're going to start a clot, somebody's going to get pregnant, somebody's going to do a shot, decide they're going to try ecstasy, I don't know what they're going to do, but all of them simultaneously. And my 17-year-old generally like rolls his eyes and he's like, you need supervision more than I do, and he's probably right, because he's like the most rule follower kid I've ever met in my life. This is the kid who, like, he won't let us sneak water bottles into the movie theater. No. He's like, it says no outside food or drink. I'm like, well, if you want to pay $4.50, you're gonna do it.
Oh
So that's his personality.
My mom was always that loud lady with the food in her purse. Hear her like start to open it. It's like, oh god, no.
So platelets. If platelets congregate for any reason, they are likely to start a clot even if there isn't a damage. And they are more likely to congregate when you have trouble with blood flow. So can you see why clots are far more likely to form in veins than arteries?
Yeah.
Yeah, because the blood flow is way slower and lower pressure. Okay. And then if your vessels are lumpy, think arteriosclerosis, atherosclerosis, right? Okay. If your vessels are lumpy or narrow, then you're more likely to catch a platelet, right? And then add all of that to like inactivity patterns. So, for instance, you've got a person who has a poor diet and exercise, right? So their blood vessels are going to be more likely to be lumpy and a little more likely to be narrow. And then they decide they are going to take a flight to Australia, right? And then they're sitting. Well, first of all, which vessels have the lowest pressure and the slowest flow in your body? The veins in your legs specifically. And now you sit for 18, 22 hours. Yeah, they do now. They didn't used to, but then somebody had a heart attack or stroke and then sued them, and now they warn you about it. So what happens is the blood flow generally has trouble, but then you're also sitting around and you're being inactive. And you're not going to have a heart attack or a stroke on the plane. You're going to have a heart attack or a stroke after you get up within about 2 days, right? And then what happens is those clots that form, they start circulating. That's not going to be a problem in an otherwise healthy person because your blood vessels are nice and open. Your coronary arteries are nice and open. Do you remember those coronary arteries that I showed you that were about 90% blocked? Now what happens is with that combination of arteriosclerosis plus throwing a clot, that's when you have a heart attack. Or if it happens in the brain, you have a stroke or a pulmonary embolism, right? So that's thrombosis. But with most of us, if your blood vessels are nice and open, there are mechanisms that will slowly break down the fibrin. You just have to have time for that to occur before it actually causes a blockage and kills the tissue downstream. So that's what thrombosis is. So when a clot starts to move, it's called an embolus. When it's still, it's called a thrombus. Okay, so it can cause an embolism. Go ahead.
So what is it that causes clots in women over 35 when they take birth control?
Estrogen. High levels of estrogen increases your likelihood, and it's all women.
Yeah.
But it's just that over 35, you're more likely to have narrowed vessels, so it becomes more of a problem. So high levels of estrogen increases your likelihood of clotting, and that can happen with birth controls. It also happens with pregnancy, of course, your estrogen levels. So, you know, I have a heart problem, and since they knew that I had a heart problem, and mine's just like because of pathogens when I was a kid, the entire time I was pregnant, I had to shoot myself up with anticoagulants, like twice a day, the entire time I was pregnant. because the high levels of estrogen increase your likelihood of inappropriate clotting. So, and I had atrial fibrillation, which makes your blood just sit, right? So that in combination with high levels of estrogen increases the likelihood. But we both lived and it was fine. The other biggie with increasing your likelihood of clotting is smoking. They think that this is the relationship. I haven't looked to see if there's new data on this, but they think the relationship with smoking and clotting is that oxidized cholesterol sticks to the walls of your blood vessels, okay? And smoking zaps antioxidants from your system. So the cholesterol that you take in, everything sticks. So like your vessels are way stickier when you're smoking. And then of course you can increase your likelihood by doing things like being an IV drug user, right? And then that makes your vessels all lumpy, right? And scar tissue formation. So there's a bunch of things that you can do. But the biggest risk here, really, that's a big deal to us, that's totally preventable, is the American diet and exercise pattern. And the American diet and exercise pattern is high levels of sugar, high levels of fat, and it's the combination of sugar and fat that really gets you, especially animal fats. Sugar and cholesterol are the big combo. In combination with low levels of exercise, because exercise actually cleans out your blood vessels. And that is really the American diet, right? That's the American activity pattern. High fat, high sugar, low exercise. And so then it's, damn near inevitable if you add any other factor in there that you are going to end up with arteriosclerosis or atherosclerosis. Go ahead.
So they don't think that it's the smoking interacting with the cholesterol itself, it's just eliminating the antioxidants?
That is the current level, that's the current knowledge that I am aware of.
Okay.
But smoking also increases your blood pressure. So that causes a little bit more damage to your vessels. I haven't seen another path. And at that point, by the way, the research and the conclusion was relatively preliminary. They know it occurs, and they're trying to figure out the link, and they think that the link is antioxidants. So, okay. So with that, let's just briefly give you guys a little intro to the lymphatic system. And the lymphatic system, the reason I want to do the lymphatic system with you guys is that even smart people who've had anatomy before have usually not had the lymphatic system. They usually don't know where the structures are. And I don't know why. I think it's kind of difficult to teach. The terminology is a little, and it's also a little difficult to understand the significance. So my objective is not going to get you guys a really deep understanding of the lymphatic system, but I want you guys to have a tour of it so that when you meet it in the future, you'll understand. It's definitely got clinical significance because it's really, really intimately involved with your immune system. As far as the anatomy of the lymphatic system, you don't ever see it because the lymphatic system is a series of really low pressure tubes that are relatively narrow, which means they flatten. So if you're looking for them, they look just like connective tissue, and they're even thinner walled than a vein. So you would really have to know what you were looking for in order to find them. It's a series of tubes. They run parallel to your venous system, and they have nodes all along them. So the lymph nodes. Now, I know you've heard of lymph nodes, and you've probably even felt your own lymph nodes when you get sick. But that's not the only places they occur. They occur intermittently along the tubes. They're all through here. The place that you may have felt them is the submandibular lymph nodes when you get sick, or sometimes these lymph nodes up here. Sometimes here. Sometimes when people get sick, they can feel them right here. It kind of depends on the inherent size of your lymph nodes and how superficial they are, whether you can feel them when you get sick. It doesn't necessarily mean that yours are particularly worse than anywhere else. It means just the size and stuff. So there's a lot of variability between the sizes of lymph nodes. They can be like pinpoint size up to like kidney bean size, really. So what's happening here is two things. This is part of your immune system. The lymphatic system is the primary place where those lymphocytes that we learned about, that's where those circulate, okay? Lymphocytes circulate through there. And the lymphocytes, remember, are the smart part of your immune system, the part that remembers infections and makes antibodies and makes cells that are prepared for the next infection, right? So it's going to be really important in that. So the red bone marrow that makes the lymphocytes is actually considered part of the lymphatic system, right? In addition, you've got some other structures like the spleen. Most people don't know what the hell the spleen does. You'll figure it out today. And your tonsils are actually part of your lymphatic system. And you know what happens to your tonsils when you get some kind of respiratory infection, at least potentially, right? They increase in size. They increase in size because there's a whole bunch of immune activity in them during that particular time. Then there's some other structures that are going to be part of it. So we'll talk about its immune function in a decent amount of detail in just a second. But the other function of the lymphatic system is more like mechanical function of the lymphatic system. So remember when you are sending blood through a capillary bed, you're sending blood through a capillary bed, what happens is you lose fluid, right? And I told you that you lose fluid as you go through a capillary bed, but you bring 90% of it back up by osmosis. You push it out by filtration, bring it back by osmosis, right? Okay, so what about that 10%? You can't just consistently lose 10% of your blood volume every time you go through a capillary bed. If you did, you would actually look like the Michelin Man given about a day, right? As a matter of fact, you lose about 3 liters. You only have 5. You lose about 3 liters of fluid into the interstitial fluid per day. So how do you get it back? The way that you get it back is these vessels gather it up from all of the capillary beds, and they're running up toward the subclavian veins, and eventually they dump that excess fluid back up in the subclavian veins. So immune function and also draining excess tissue fluids back into your cardiovascular system. Does that make sense? Okay, and as they're doing that, they have a lot of immune functionality as well. So what happens if you get blockages in these locations? So breast cancer, when someone has a mastectomy because of breast cancer, the lymphatic system is a really common route of metastasis for cancers. So you may or may not know that, but what happens is if somebody has a mastectomy, they don't just take the breast tissue, they also take all of the lymphatic vessels and lymph nodes from this area. But that, of course, causes a problem with draining excess tissue fluid, right? So if someone has a mastectomy on the left side and takes all the lymphatic tissue on the left side, what happens to this left arm? It swells, right? And a lot of times what they do is they put compression garments on it to help with the swelling. As soon as you take the compression garments off, the swelling comes back. Eventually, by the way, your lymphatic tissue is regenerative, so you can actually get it back. But so that is called lymphedema, when you actually get a swelling because of excess lymphatic or poor lymphatic flow. It's called lymphedema. Now, the worst examples of lymphedema are elephantiasis. And with elephantiasis, what happens is there is a parasite that is communicable from person to person because the eggs are circulating in your bloodstream. If I were infected with this parasite, the eggs would be circulating in my bloodstream. And if a biting fly or mosquito bit me and then bit you, because we live close to one another in the same village, does that make sense? Then what happens is it's communicable. It's not directly transmissible, but it's carried by these biting flies and mosquitoes. And the problem with elephantiasis is that when that organism starts to go through its life cycle, the larval stage of this little worm actually likes to hang out in these vessels. And then it grows in the vessel. And as it grows bigger, it blocks drainage in the vessel. Does that make sense? And so you can then get elephantiasis. So you see all this excess tissue fluid that's building up in this vessel. So this is treatable. The treatment's kind of nasty because it is an animal parasite and you are an animal. And they have to pretty much poison it out of your system, right? So it's not like you can use an antibiotic and you don't have a cell wall, so you're cool with an antibiotic, except it might upset your stomach. You can't use an antiviral that doesn't have anything to do with your cells. You have to use something that actually damages animal cells, and you're an animal. So the treatment is pretty nasty. It does work. But the problem is, in addition, you have to treat multiple people in order to get this out of the system, right? You have to treat not just the people who are infected, but the people who might be infected. Otherwise, it continues to go from person to person. So that's elephantiasis. Here's another picture of elephantiasis. Okay, and then this one is a pretty famous picture of elephantiasis of the testes. Yeah, there's a, I couldn't find it. There's this pretty famous picture of elephantiasis of the testes. And it is, of course, more common in areas where there's lots of moisture because a lot of it is mosquito-borne, okay? Why testes, why these places? If you have fewer alternate routes of circulation, then the blockage tends to cause, of course, more lymphedema. I have a picture in which, like, in a single village, there are 4 men who have such massive elephantiasis of the testes that they actually can sit on their testes and use them as like a hippity hop. So, yeah, it's like, I mean, I don't think there was any bouncing, but still. So, we'll talk about the function, the HIV and AIDS function with the lymphatic system in just a second. So, we'll come back to HIV and AIDS when we talk about the T cells and the B cells. So, I'm going to show you a short little video after we talk about the other functionality, and then we'll pick up here next time. The other thing that the lymphatic system does is sometimes proteins leak out via filtration, but you can't pull them back up by osmosis because osmosis only moves water, right? So leaked stuff that you can't get back up by osmosis goes back into the lymphatic system. In addition, and this one's kind of interesting, you will look this week in lab at least one lymphatic, starting Friday, in lab at least one lymphatic structure. One of the cool and weird things about absorption of nutrients is that fats get absorbed differently than other nutrients, than like carbohydrates and proteins and amino acids. So you absorb carbohydrates and amino acids and most vitamins. You absorb them into the bloodstream. But with lipids, you absorb them into the lymphatic system directly. So the important part of that is that means if they go into the lymphatic system and they have to percolate through the lymphatic system before they get dumped into your subclavian veins, that means that they have bypassed hepatic portal circulation, right? That means that any lipid-soluble toxins, any pathogen that moves into the lymphatic system gets around practically through the body before it goes through hepatic. I mean, it will eventually go through hepatic portal circulation, but not the first round through hepatic portal circulation. That means that lipid-soluble stuff escapes surveillance by the liver. It's more likely to be toxic. It's also harder to clear from the liver and the kidneys. So lipid-soluble substances tend to be a little harder on your body. The other interesting thing about that is that means that right after you eat, of course we know that your blood glucose level goes up. But what about your fats? If you looked at your lymphatic fluid right after you eat, your lymphatic fluid is all milky and viscous, especially if you have a fatty meal, like right after Thanksgiving. Your lymphatic is like greasy. And then eventually it dumps it into your bloodstream. So you get like this period in which you don't have fats in your bloodstream and it gets all dumped into your bloodstream, which is kind of gross to think about. Okay, so we will do this and that next time. So I'm just going to show you a short little 4-minute video about lymphatic system and then I'll let you guys go. Hold on. Or not. Is it this one that doesn't? Hold on. Audio off, text on. Oh. What?
The very top by the on the left versus off.
By the browser address.
Yeah.
By the browser address? Oh no, that's an ad blocker. Okay, I'll just post this for you guys. This thing doesn't do audio apparently, so I'll post this for you guys to give you a little intro, and then we will pick up on Thursday after your test. No lymphatic test.